right, so I'm sure everyone here is familiar with the classification in Chiari malformation, and I'm not really going to repeat that. I'm just going to emphasize that there are different levels of severity. Is this better, guys? Okay, so I'm just going to emphasize that there are different levels of severity. In level 1, essentially, what we have is a deformity of the bone and brainstem structures. And in types 2 and 3, in addition to the deformity, we have a bone defect resulting in meningocele and basically outpushing of the neural structures. Now, type 4, I am convinced that phenotypically speaking has nothing to do with Chiari malformation, but as you know, these classifications were given by the old school neurologists who wore bow ties. So essentially we have to concur, we can't object to it, but I personally am convinced that type 4, phenotypically speaking, has nothing to do with Chiari malformation. All right, so what went wrong with brain formation? Why there is a malformation? I want to emphasize that formation of brain is orchestrated by very complex cell signaling pathways. And what is of interest in the case of Chiari malformation is a reciprocal development, reciprocal relationship between the development of the brain and the bone structures that are covering it. So somehow these cell signaling pathways are impaired, resulting in a deformity and abnormality involving both. So in the case of type 1, what we have is a small occipital bone and some deformity in the posterior fossa structures that are tightly packed. Now, to illustrate this a little bit better, I'm going to show you this picture of a developing embryo. The entire nervous system is basically made from a tiny tube on the back of the embryo. And this tube folds upon itself several times and ultimately produces the hemispheres, the brainstem structures, cerebellum, and down below the spinal cord. Now, in the center of this tube, we have the stem cells. These stem cells multiply and create 3 different cell lines that produce the neurons, the ones that produce the myelin-producing cells, and ultimately the supporting cells or glial cells. These cells multiply in the center, and then they migrate out, and this happens mostly during the first 3 months of gestation, and afterwards, during the most complicated process you can imagine, these nerves basically produce extensions, dendrites, and axons, and create neuronal networks and make us who we are. So basically what has gone wrong in the case of a Chiari malformation is disruption of the cell signaling pathways in this part that result in lack of growth in the bone, occipital bone, and some deformity of the spinal, of the posterior fossa structures. That's of course just in Chiari 1 malformation. In types 2 and 3, as I emphasized earlier, what we have is more than just the deformity. There is lack of bone formation such that the meninges stick out and the neural structures stick out with it. All right, talking about the genetics of Chiari malformation is quite challenging because not much is known about it, and even the most exhaustive search of the literature and databases would give you not much more than saying that it is sporadic. Basically, there are no known genetic mutations that have been identified. And right now we're talking about Chiari type 1. There's a naturally occurring animal model which occurs in a species of dogs that have been used for studies. And there's a great deal of familial clusters that have been reported. So these are parents with children that are affected, or twins or other siblings. Now, in the case of Chiari type 2, again, the most exhaustive search of the literature and the databases tells you that it is sporadic, that there are rare reports of familial clusters, and there is really no single gene that has been identified. Of course, in type 2, as I emphasized earlier, it may be associated with basically neuronal migrational defects. So these children may be more severely affected in terms of cognition and development. And in an animal model, in the mice, Chiari type 2 has been mapped to a portion of the neural crest that basically deals with mesoderm. And mesoderm is part of the embryo that produces the soft tissue, the bone, structures that in essence cover the brain, and hence the neural tube defect that occurs in these people. So to add more to my discussion, I searched the neural tube defects, and as you may have heard, these are complex traits, and they're multifactorial. They're very common, and next to cardiac defects, in fact, are the most common type of birth defects in children. Well, there is a great deal of environmental factors that cause or may cause neural tube defects. And as neurologists, we know quite well that using anti-epileptic medications is one of them, in particular using DEFECTS, other types of medications. And one very important factor is deficiency of a vitamin we call folate or folic acid. Maternal diabetes and high BMI in the mother, they may all be contributing factors. And I came across one study, a whole genome association study that basically looked at the variations in genes involved in folate metabolism that may be associated with neural tube defects. So basically the most common form of neural tube defects is spina bifida occulta, so that's completely asymptomatic. There is a bone defect, but not much more than that, and even that you have to find with very careful examinations or x-rays. But in the more severe forms, there is protrusion of the meninges and the neural elements from the area of the bone defect, and of course that can happen in the lower end at the level of the spine or at the upper end at the level of the back of the head, brain. And again, there is no single gene that has been determined in cases of neural tube defects. As I was saying earlier, these are complex traits, and there is a combination of environmental factors and genetic causes that play a role. And of course, in cases of Chiari malformation type 2 or 3, these co-occur. So the genome-wide association studies basically have suggested presence of 4 genes that are potentially associated with neural tube defects. And again, these are probably susceptibility factors and not causal genes. And I would explain that in a little bit. So what genome-wide association studies are, this is a method that involves studying large numbers of patients. So as you may know, the start of genetic studies in the 1970s was basically linkage studies. And what they would do is that they would get a family with some affected and unaffected family members. They would get a sample of blood, they would get the DNA, and then they would have some kind of an educated guess about which markers they should test. They would check for those particular markers in the affected and unaffected family members, and they would decide if those markers co-occur with the disease. Now, imagine doing that, those family linkage studies, in a much larger scale. So in the case of genome-wide association studies, they need large number of patients that are affected, and of course you would need to have a pool of unaffected people. You extract the DNA either from a blood sample or a swab from the mouth, and you basically extract that DNA and import it into a chip, and the computer systems, basically the high throughput sequencing methods, do the rest for you. Now again, you have to have an educated guess about which markers you have to check for. And fortunately, after completing the Human Genome Project and HapMap project, making this kind of educated guess has been a lot easier. And then you basically try to determine if there is certain markers that occur with the disease and not in the normal subjects. And what you come up with here is really not causal genes, it's susceptibility factors. So again, susceptibility factors are genes with very tiny effect. Therefore, having one of them often does not cause the disease. You have to have multiple susceptibility factors. And that's the case for many disorders with complex genetics. So for example, in the case of Alzheimer's disease, in the case of Parkinson's disease, you don't really, in majority of cases, you don't have a single gene. You receive susceptibility factors from your parents. So if you're unlucky enough to have 5 susceptibility factors from your mom and 5 from your dad, then you're going to get Alzheimer's. And that's why in majority of these cases, you cannot identify a single, like a very simple pattern in the family. Did your father have Alzheimer's? No. Did your mother have? No. So why do I have it? It's because it's not a single gene disorder. It's complex disorder, and it's inherited by, through the susceptibility factors. So I was able to find only a single study of genome-wide association studies in Chiari malformation. And this was published in the American Journal of Medical Genetics in 2006. And they recruited 130-some families with 71 affected members. And they basically scanned the entire genome for 10,000 markers. And they found association with 2 loci. One of them is on chromosome 15. It's called fibrillin gene. And the other one is on chromosome 9. And again, these are susceptibility factors and not necessarily disease-causing genes. So in conclusion, based on what we know so far, Chiari malformation is sporadic. There is not a single gene alteration that has been identified. There are reports of familial clusters, and they, in terms of pattern of inheritance, they seem to follow autosomal dominant or autosomal recessive patterns. In the case of neural tube defects, there are more information about potential susceptibility factors, and of course they co-occur with types 2 and 3 of Chiari malformation. And the genome-wide association study in Chiari malformation has suggested 2 markers. Now at the end, I'm going to add my personal anecdotal experience. I personally believe that the familial clusters are a lot more frequent than what's reported in the literature. And I observed several cases of familial clusters per year. So I think it's a lot more common than what we think. Personally, what I have observed has always been involvement of a mother and a son. I have never seen inheritance from mother to daughter or from father to children. And this pattern, in my opinion, is suggestive of an X-linked transmission. And ultimately, of course, the genome-wide association studies are helpful for determining the susceptibility factors, but in order for us to come up with the actual genetic cause, they would need to do exome sequencing or whole genome sequencing studies. That was the end of my presentation, and I'll be happy to entertain questions, possibly at the end.